Okay, welcome to the first video of Unit 3 VCE Biology, the Signatures of Life. This video will be using the cell structure PowerPoint, and proviso with all of my videos that this is certainly unplanned and unscripted and will most likely be unedited, unless of course I work out how to do editing. So in which case please go easy on me, and any feedback is always welcome. This PowerPoint in this video, cell structure, covers things that are not necessarily explicitly covered in the study design, but they're things that are, I guess, implied that you need to understand for VCE biology. So we'll be looking at things like the different types of cells, prokaryotic and eukaryotic, what defines those types of cells, and then some of the specific organelles that we'd find within those different cells. Okay, I'm also going to be skipping through quite a number of the slides on here. Of course, the PowerPoint is available to you, so you can have a look through those, and we'll even check some of the things out in class as well. But just probably don't have time to cover everything in this video. So let's skip through and start with looking at commonality in all cells. And this means, what are some defining features that we'd find in every single cell, regardless of whether you're a bacteria or a human or a plant. Every single cell is going to have these different things. So every cell will have a cytosol and a cytoplasm. Two very similar words, but referring to slightly different things. And this is an important distinction to make. We need to know what a cytosol and cytoplasm is. Of course, the cytosol is the fluid, the intracellular fluid that we find in a cell. It's the thing that bathes all the organelles and bathes the nucleus. And it contains largely water, and it's going to have some dissolved ions in there as well, and probably a lot of proteins as well. But it's the fluid. The cytoplasm is the cytosol plus all of the organelles that it's bathing, except for the nucleus. So, it's a strange distinction, but it's an important one to make. It's everything that's in between the cell membrane and the nucleus. So the cytosol and all of those different organelles, all the organelles that we'll hear about a little bit more in this video. So the distinction between those two is really important. The third thing on here is the cytoskeleton. The cytoskeleton is, I kind of refer to it as cellular scaffolding, because in many ways it acts like a scaffolding. It helps with the structure of the cell, so it helps to keep everything rigid and in its place, I guess, but it also provides a good passageway for things to move around the cell. It's almost like a transport system within the cell as well. And we find different types of cytoskeletons in prokaryotic and eukaryotic cells, but we do find cytoskeleton in all cells. Notice that each three of these words as well start with the prefix cyto. No surprise, cyto meaning cell, something that's very common when you're looking at features of cells. Okay, every cell has also got a cell membrane or a plasma membrane, different words for the same thing really. We know a lot about the cell membrane already. It's that barrier that controls the movement of things in and out of the cell, that separates the intra to extracellular environment. We'll look at that a lot more detail in the future. Some cells have got a cell wall. Plant cells we know have got cell wall, fungi, but not all cells do. Certainly animal cells don't have a cell wall. So it's not a feature of all cells, but all cells have a cell membrane. All cells. Okay, we also find DNA in all cells, feature of all living things. You've got to have DNA if you want to be alive. Just the way it is, I'm so sorry. It might be in different forms in different cells. So on the this side here, we've got probably a eukaryotic cell with long linear chromosomes. The DNA is in long linear chromosomes. Whereas on the other side here, we've got a prokaryote, maybe a bacteria, and it has a single circular chromosome. So different structure, but still each of those chromosomes contain DNA. Now, DNA we know codes for proteins. It's the genetic material that codes the proteins that cell makes. But you've got to have the equipment to turn that DNA into an actual functional protein. And the equipment that does that is a ribosome. So if you've got DNA, there's no point having DNA unless you're going to have ribosomes as well. So all cells have ribosomes to turn that DNA or to decode that DNA into a protein. So all cells have commonality, has things in common, but cells also have things that are different. We've heard about eukaryotic and prokaryotic cells before, and there's lots of differences between them, but these are probably some of the key ones that are worth noting. So eukaryotic cells are the cells that have a nucleus and have a whole bunch of other organelles that are bound by a membrane, bound by that same stuff that the plasma membrane is made out of, a phospholipid bilayer. So eukaryotic cells, of course, if we look down the bottom here, this branch here goes to fungal cells, plant cells, protists and animal cells. So we are made up of eukaryotic cells. All of our cells have a nucleus and membrane-bound organelles, and that's a really key defining feature of eukaryotic cells. We've got multiple linear chromosomes, and they're relatively large cells, relatively because cells are still pretty small, but they're much larger than the prokaryotic cells. So prokaryotes are, well, there's bacteria are kind of the main group that we would worry about when it comes to prokaryotes. You can also look at archaea, but bacteria for our purposes are prokaryotic cells. And they don't have a nucleus. Their DNA is just in a region within the cell. It's not bound inside a nucleus. And they don't have any other membrane-bound organelles. They have organelles because they've got ribosomes, but ribosomes aren't membrane-bound. So they don't have chloroplasts or mitochondria or Golgi or endoplasmic reticulum or anything like that because they don't have any membrane-bound organelles. They've also only got a single circular chromosome where we find their DNA, and they're small. They're very, very small, the smallest living things, significantly smaller than eukaryotes. So there are similarities in cells and there are differences. The first difference is prokaryote versus eukaryote. I think we'll have a look at this one in class with amazing animations on there we can run through. But then we want to delve down into the eukaryotic cells. Prokaryotes, there's not much more to them. They're pretty straightforward. We'll look at them every now and then. But eukaryotes, we can branch out a number of different types. Now, we'll want to know a couple of things about fungal cells and protists, but for the most part, the key things that we want to know about are the differences or the similarities and differences between plant and animal cells. That's where the real business is at. That's where the money's at. This is what we want to know about. And things that are common to all plant and animal cells are, well, the things that are listed here, the plasma membrane, the cytoskeleton, we've already looked at those, nucleus, nucleolus, mitochondria, list goes on. Let's have a look at some of those and then we'll have a look at some of the differences between them too. So plasma membrane we know a bit about. We're going to come back to it in a lot more detail when we deal with movement across the membrane. So I'm going to leave it for now just to save us some time. Ribosomes I spoke about a moment before as well. Ribosomes can be floating around in the cytoplasm on their own. They can also be stuck to the endoplasmic reticulum, where it's called the rough endoplasmic reticulum. They're made up of two things. They're made up of ribosomal RNA. When we look at nucleic acids, we'll see there's 3 different types of RNA, one of them being ribosomal RNA, and they're made up of some protein as well. And their function is the synthesis of polypeptides. So these are the guys that turn DNA, or we'll look at the process in more detail, but essentially these are the guys that decode the DNA and turn it into a functional protein. Maybe that's a hormone, maybe that's an enzyme, but they turn that genetic code into a polypeptide or a protein. They're essentially similar, the same things. So ribosomes, common to all cells. The nucleus, found in all eukaryotic cells. It is, let's see, so it's a membrane-bound organelle, so it's got a membrane, and we give that membrane a special name. We call it the nuclear membrane, no surprise. And that membrane is not completely enclosed. There's lots of little holes in that membrane, or nuclear pores, we will refer to them too. And they'll be important when we look at how DNA gets decoded. Inside that membrane, we find the DNA, the genetic material. In the form of chromosomes or another material called chromatin, we find that DNA. So when it comes to the function of the nucleus, a lot of the time we might simplify it and say it's the control center of the cell. That's what it does. It controls the cell's activities, which is true, but it's not getting down to the specific nitty-gritty of it. When we talk about the function, we want to say that it contains the cell's genetic material, and I've got most of the genetic material because we'll see there's genetic material elsewhere in the future. But really, you could say that it contains the cell's genetic material, or it contains the cell's DNA. That's how it manages to be the control center. Within the nucleus, we find the nucleolus, this darker patch in the middle here. Don't need to know much about the nucleolus. All we need to worry about is that it synthesizes ribosomal RNA. And we said ribosomal RNA is found in ribosomes. Where does it come from? It comes from the nucleolus. That's all I'd worry about with that. The rough endoplasmic reticulum mentioned before as well. It's an extension of the nuclear membrane. So that nuclear membrane, which is a phospholipid bilayer, can branch out and form this endoplasmic reticulum substance as well. Its structure is that it's this membranous tubules, so it's kind of like a passageway, I guess, of all of these little membranes, and you can see the gaps in between the membranes here, these little passageways that get formed. And it's rough because it's studded with ribosomes, lots of little ribosomes stuck onto the outside of it. Its function has a number of functions. It transports proteins throughout the cell, just within the cell itself, and it can do that by proteins being able to move through these channels to get to another area of the cell. And it also is able to release transport vesicles, so little vesicles can bud off and move to another part of the cell as well. It's also involved in the synthesis of proteins because it's got an association with ribosomes. It helps to synthesize them, but then it also helps to change them a bit, to modify them, to fold them, to turn them into different types of protein molecules. We'll look at that more in the future. So the rough endoplasmic reticulum we find in all eukaryotic cells. We also find the smooth endoplasmic reticulum in all eukaryotic cells. And this is something that is connected to the rough endoplasmic reticulum. This membrane will extend on and become the smooth endoplasmic reticulum. And its function is quite similar to the rough as well. It's obviously not involved in protein synthesis because it doesn't have any ribosomes on it, and that's why we call it the smooth endoplasmic reticulum. But it does transport materials throughout the cell through these little channels that we find in the membrane. It's also involved in the synthesis of lipids. That's oils, phospholipids, steroids, a whole range of different lipids there. Where do you make them? You make them in a smooth endoplasmic reticulum. Okay, from those endoplasmic reticulums, if we wanted to move a functional protein or substance out of the cell, if we wanted to export it from the cell, we would need to utilize a Golgi or a Golgi apparatus or a Golgi complex, or a Golgi body, all the same terms that refer to the exact same structure. The Golgi acts in many ways in association with the endoplasmic reticulum. Its function, let's look at its structure first. So it's these flattened sacs of membranes, and if they're in an electron microscope image, they look like down the bottom here, these flattened discs, and if we look at the diagrammatic representation up there, we can see them. Its function is to receive transport vesicles or transfer vesicles from the endoplasmic reticulum, and these vesicles will be containing things like proteins. It might modify them a little bit more when those vesicles fuse with the Golgi. The contents then get released into the membrane sacs. It might modify the contents a little bit more, and then it will package them back up and transport them. This case, though, it's going to transport them out of the cell. The Golgi's function is to transport materials out of the cell. So these vesicles will then head off to the plasma membrane to be released via exocytosis. Speaking of vesicles, we've looked at them a lot before, little membrane bound sacs essentially, which are really important for transporting things throughout the cell. Vesicles are very similar in their look to vacuoles, and vacuoles are often a little bit larger than vesicles, and especially if you're a plant, then we'd expect a plant to have very large vacuoles. And vacuoles form a slightly different function. They're not there to transport things around the cell necessarily. They're often for the purpose of storing things. So in animal cells, we might find food vacuoles that store food, surprise, surprise. In protists, contractile vacuoles, which help to pump water in or out of the cell. And in plants, importantly, we find a central vacuole. And the central vacuole in plants can be really large. It can take up the majority of the cell and push the rest of the cytoplasm right out to the edge of the cells. It contains a lot of water and some salts as well, and it's the thing that gives the plant cell its turgidity. It's the thing that keeps the plant nice and strong and standing up without the aid of any type of skeleton. Okay, at my 15-minute limit here, so I think we'll stop this video here and then we might come back for part 2 momentarily.